Hello learners, myself Dr. Poonam Patil. Today I am going to discuss about Digitalis. Digitalis is a well-known drug which is from the cardiac glycoside. So synonym for this drug, there are various synonyms for Digitalis leaves, foxglove leaves. Biological source, it consists of dried leaves of Digitalis purpurea, belongs to family Prophylleraceae. Actually, this drug is dried temperature below the 60 degrees Celsius immediately after collection of leaves. Then it also requires some precautions, right? Because this drug contains 0.3% of total cardenolides, which generally contain digitoxin. So special precaution is required. So it is immediately dried, not exceeding 60 degrees Celsius, and it is stored in moisture-proof container. So this is the physical appearance. This bears very beautiful flowers. Then for the this drug firstly reported in Russian Pharmacopoeia in the year of 1860, and afterward William Withering elaborated the work on the digitoxin and reported in some special books. Then for the cultivation purpose, actually this is biennial perennial herbs which having 1 to 2 meter height, so it requires the altitude about 1600 to 3000-meter altitude. Then temperature condition 20 to 30 degrees Celsius. Rainfall 30 to 40 centimeter per annum. And this drug is simply propagated by the seedling method. Broadcasting method is generally preferred. For the soil condition for this cultivation, calcareous, acidic, sandy soil required for favorable growth. Then these seeds are very small in size, so 100 seeds weigh 40 to 70 mg, and for per hectare it requires 1 to 3 kg seeds. Then these seeds are mixed with fine sand and then sown in nursery bed in the month of March and April. And after it develops the young seedling, then it is transplanted into the field in the month of September, November. And this crop is manured and kept free from weed. Then plantation done in twice a year. So plant bears the flowers in the month of April and it is followed by the fruiting. At that time, the leaves are collected and it generally bears the rosette leaves. And in second year, it bears sessile leaves. So leaves are collected in second year in the afternoon during August and September when two-thirds of flowers are fully developed. And discolored leaves are rejected. After collection, leaves are immediately brought to drying center and dried in vacuum dryer at temperature below 60 degrees Celsius till the moisture is not more than 5%. So dried leaves are packed in airtight container with suitable dehydrating agent. If the leaves are dried above 60 degrees, potency is lost due to the chemical degradation, and those are the glycosidic linkage, it is degraded, so it lost the potency. So activity of leaves is due to the glycoside and it should be preserved at below the 60 degrees Celsius. So presence of moisture and enzyme, that is digiphosphates and oxidase, which cause deterioration after the hydrolysis. Then next is macroscopy of this drug. So these leaves are dark grayish green in color, odorless, distinctly bitter in taste, and size is about 10 to 30 centimeter in length, 4 to 10 centimeter in width, and shape ovate lancet to broadly ovate. So this is the type of leaves and which having dented seriate margin. Then venation pattern, mostly pinnate type of venation pattern, and it is very soft to touch both the side, upper as well as lower side. Base is dentate, apex subacute, and margin is great and dentate. Then microscopy. So this is the dorsiventral type of leaves because of palisade tissue are present only one side. Then it bears anemocytic epidermis cell containing anemocytic stomata, both the surface, water pores also present. Then covering as well as glandular trichomes are present on both the sides, which both are the multicellular in nature. So glandular trichomes are short, unicellular stalk, and unicellular or bicellular heads. Covering trichomes, they are uniseriate with collapsed cells. Then these digitalis are free from calcium oxalate crystals and sclerenchyma, but it containing numerous numbers of starch grains in the parenchymatous tissue and collenchyma present on both the sides. As well as below the vascular bundle, there is the presence of endodermis, then lignified pericyclic fibers also present. So this is about microscopy of digitalis leaves. So here figure shows the complete uh, means details of the tissue which is present in the digitalis. After that, chemical constraint. So digitalis contain 0.2 to 0.45% of both primary and secondary glycoside. Primary glycoside like purpura glycoside A and B, then glucogitaloxine, then secondary glycoside that is digitoxin, G-toxin and G-taloxine. So primary glycosides are less stable, less significant than secondary glycoside. Purpura glycoside A, B are the main principal constituent of the fresh leaves. And this is about the uh, means uh, presence of means formation of secondary glycoside from purpura glycoside A and purpura glycoside B. And this is the difference between the structure getoxygenin and digitoxygenin. Actually, this uh, detoxin containing one glucose molecule and G-toxygenin containing means after hydrolysis of G-toxin it containing digitoxygenin plus 3-digit oxo sugars. Then other than this glycoside it also containing two saponin glycoside that is digitonin and G-tonin and enzymes also present in this digitalis leaves. For the identification there are three identification tests for uh, cardiac glycoside, that is killer Killarney test for the digitoxo sugars, then legal test for the cardinolites and budget test also for the cardinolites. Next is uses. So this drug specially recommended for congestive heart failure. Then how it shows the it potentiates the action means firstly it blocks the sodium potassium ATPase pump of cardiac muscle. After that it ultimately increases the intracellular concentration of sodium ion. It increases the release of calcium ions from the sarcolemma, so result in forceful contraction of myocardium. There is the greater output per bit and complete emptying of heart because of the flushing of edema, and delayed the circulation is improved due to the flushing of this edema. And this is about the how this drug increase the positive ionotrophy effect in sodium potassium ATPase pump after blocking of sodium potassium ATPase pump. So here digitalis simply act to block the sodium potassium ATPase pump and then it increase the positive inotropic effect. So it increase the excitability of cardiac muscles and produce more powerful contraction and improvement through the kidney result in diuresis and loss of edema. So it is effective in congestive heart failure to increase cardiac output and to relieve venous congestion. Hence it is used as a cardiotonic. And major disadvantages of this drug, it has a cumulative effect. So administration of drug should be under strict medical supervision. Then storage, it should be stored in well-closed container in cool place, and leaves do not contain more than 5% of moisture because it causes destruction of glycosides. Then adulterants for this drug, the mullein leaves, which is obtained from Verbascum thascus, and it is densely covered with a large branch woolly hairs. So this is the difference. And Conferry leaves, second one, that is Symphytum officinalis, belongs to family Borginaceae and it can be detected by the presence of multicellular trichomes. And third adulterants, that is primrose leaves. And botanical source for this primrose leaves, that is Primula vulgaris, family Primulaceae, and it is detected microscopically by the presence of vinicerate covering trichome. So this is about adulterants. Next is allied drugs. So allied drugs for this Digitalis lanata, which is called white foxglove, and synonym of this drug woolly foxglove leaves, botanical source that is Digitalis lanata, part use that is Digitalis dried leaves, family Scrofulariaceae. So this is Digitalis lanata. Then it also containing same constituent that is Digitalis digoxin and other glycoside, then digoxygenin and digenatigenin. So this is the specific to digitalis, while other present in digitalis purpurea. So this is about the structure of chemical constituent which is present in digitalis lanata. So primary glycoside, that is lanucleotide A, B, and C, and after the hydrolysis, it is converted into digitoxin, B converted into gitoxin and digoxin. So this also used in the treatment of auricular fibrillation and congestive heart failure, and it is preferred because it is less cumulative effect than digitalis purpurea. So this is about the chemical structure of digoxin and digitoxin. These are the species for allied species for digitalis lanata, that is digitalis lutea and digitalis thapsi. Thank you.